A very good afternoon, everyone. Thank you for joining us in this webinar. This event is brought to you by the Institute of Advanced Studies and the Graduate Students Clubs of School of EEE, Biological Science, as well as the School of Chemical and Biomedical Engineering. I'm Vivek from the School of EEE, and I will be the host for this talk on biointelligent lasers for healthcare, tiny lasers that can feel and think. Please use the Q&A feature at the bottom of your screen to post your questions during the webinar. At the end of the webinar, you will also get to interact with Prof. Chen to ask your questions directly. Do raise your hand so we might be able to unmute you so that you can ask your question directly. Today's distinguished speaker, Nanyang Assistant Professor Yu-Cheng, is a leading pioneer in the field of biolaser. Prof. Chen's research focused mainly on the development of novel photonic sensors, optical microcavity, biophotonics, bioimaging, and on-chip laser devices for next-generation biomedical applications. He has published more than 60 top journals and proceedings, with widely reported by more than 200 media outlets worldwide. His research group is devoted to inventing novel tiny lasers with programmable and biointelligent functions for on-chip disease diagnosis, neuroanalysis, and cancer detection. We're also very excited to share with you that Prof. Chen has recently been awarded the 2021 MIT Technology Review Innovator Under 35. Without any further delay, I would now like to welcome our distinguished speaker for today, Assistant Professor Yu-Cheng, to give his speech. Thank you. Prof. Chen, please.
Okay, so you guys can hear me? Oh.
Yes, Prof.
Okay. So welcome everybody, late afternoon and Friday. My name is Yu-Cheng Chen, Nanyang Assistant Professor, Nanyang Technological University. Today I would like to give a topic on biointelligent lasers for healthcare. Okay. Okay. So since the topic is called biolaser, right? So laser, we are all very familiar with laser. In terms of medicine, surgery, cosmetic, and also in industry, communication, even in military and defense. So what is actually biolaser? So the scale of laser from 1960, the first laser has been developed. And then the size of the laser and the scale of laser has been shrunk down over the past 50 to 60 years. As you can imagine, in the late days, it was a very big laser. And then to nowadays, it has become like a laser diode in a laser pen. And in nowadays, micro to nanoscale technologies has even allowed the laser to become at the sub-micron scale in the current technology now. All right, so laser has three major components. Although many people today's background may not be in optics, however, here I would like to give a very brief introduction of the three major components of a laser. Laser has a pump source, and a second component is a gain medium. The gain medium usually requires a crystal, semiconductor, or like a gas. And the third component is the optical cavity. As you can see here on the right-hand side, two mirrors can serve as an optical cavity. And then other types of cavity we will be introduced later. So these are the three major components to form a laser. So the identification of biological laser appeared in 2011. So the concept of biological laser, or I would just call it biolaser, is still very similar to the structure of a conventional laser. It has a pump, has a gain medium, and also has a optical microcavity. However, here we replace the gain material inside with biological mediums. For example, like bacteria, virus, DNA, proteins, and even living cells. We take advantage of the optical cavity to amplify very subtle changes of the biological substance inside of the cavity. As you guys may know, laser, the A stands for amplification. So here we take advantage of this cavity to amplify very tiny physical or chemical changes inside of the cavity. And then we use the laser emission as the detection or sensing signal. So this is why and how we define a biological laser. The reason why we use laser compared to the most well-known fluorescent detection is because laser actually provides very narrow line width, as you can see in the bottom here. It has a very narrow line width. It has a very sharp spectrum and very high intensity. A very unique part is it has a threshold behavior, which means that the pump intensity is nonlinear, proportional to the intensity output. So taking advantage of these unique features, a laser-based detection has very, very powerful potential for imaging and sensing compared to conventional fluorescence. Various types of optical micro to nanoscale cavities have been developed. So as I mentioned in the previous slides, there are three components. As you can see here, there are three components of this biolaser. So here, I would like to introduce various types of optical cavity. So the most famous type is formed by two mirrors. As you can see here, which we call Fabry-Perot microcavity. The light will be confined in the two mirror and forced up and down, and then it can enhance the interaction between the molecule and the light, and then therefore generate laser emission. The second type of cavity, which we often use, is called whispering gallery mode, which takes advantage of a ring or like a round sphere structure. As you can imagine, you can see the light can be confined on the spherical surface, and then it actually resonates around the sphere, and therefore it can also amplify and enhance the interaction. Many types of WGM cavity have been developed, for example, by using fiber, or even by making the center of the fiber bigger. Like this one, they call it like the micro bubble, or the micro bottle cavity has also been developed. Or the most common cavity is like droplets, taking advantage of liquid crystal, biological materials, and even oil droplets. The bottom, we show photonic crystal and distributed feedback cavity. This take advantages of like a 2D, 1D nanoperiodic, like a nano or microperiodic structure, which can also confine the light. Random scattering is also another type of common laser cavity, but actually there's no cavity. So it takes advantage of the scattering of the light to enhance the interaction. However, it is still considered as one type of a general mechanism to develop and generate laser emission. Plasmonics, finally, or metasurface, is well known over the past 10 years, which takes advantage of an evanescent field, especially by some semiconductor or like a noble metals. So this is also a very, very famous material which can serve as a cavity. In terms of biomaterials for lasing, in the past few years, for example, many types of biomaterial have been used to demonstrate lasing. For example, by using leaf, which is the chlorophyll, or by extracting the luciferin from a firefly, which is type of bioluminescence, or even other groups have used, for example, egg white from real egg, and can also serve as a biomaterial. In the bottom, you can see other groups have used vitamin B2 to serve as a gain, or also like fluorescent protein, and even the starch extracted from potatoes can also serve as a cavity itself for lasing. So here are just some general structures and materials which have been used for biomaterial lasing in the past 2 years. So the aim is to develop bio and functional laser-based sensing and imaging from the very bottom level, which is the biomolecular level, to cell level, to the network and tissue level, and finally in the human level. So this is our main goal, and in the future aspect which we anticipate in the long run. Okay. So based on the very rough idea, I hope you get the concept of what biological laser is already. Here today in this talk, I will introduce how biological laser can be applied into 4 mainstream applications, including sensing, imaging, bioinformatics, and on-chip bio devices. The first one is biosensing. Okay, so biomedical sensing, how can we actually use this laser for biosensing? So in the top, you can see as example, at the very beginning, for example, there's nothing binding to, for example, the IgG molecule. And then once a protein or maybe a cell or a peptide binds to a certain molecule, lasing or laser emission will be generated. So here in the bottom, we show the major three parameter, which we use as identification for biosensing. The first one is called lasing threshold. Lasing threshold is very unique behavior in terms of biolaser. So different substance may have different threshold. So as I control the pump intensity, I can create and generate a laser according to different threshold. The second one is wavelength. According to the right-hand side, you can see wavelength shift is also a very common behavior, which we can use to identify biosensing. The last one is by using the intensity. For example, in the center, you can see that the red one has a very sharp signal, while the green one, the green curve, is almost nearly zero. This intensity difference is very huge, which is a very important factor and feature that can be used for biosensing. So taking advantage of threshold, intensity, and also the wavelength shift, we can use laser for detection and biosensing. As we start from the application side, here we show some example of protein-based biodetection. Fiber is one of the most common structure which can be used to demonstrate biological laser sensing. We can either coat on the fiber surface, for example, to achieve a single molecule or single monolayer detection, like by detecting some thread protein or just single protein interaction, or even ELISA. The center one shows example of utilizing the hollow core fiber. So we can design and we can fully functionalize the inner surface of the fiber. So this fiber not only serves as a resonator, it can store some reagents to achieve some multifunctional fiber, multifunctional laser eventually. So this is another way of utilizing a hollow core fiber to achieve biological sensing of protein. The right-hand side one shows a conventional cavity which forms by two mirror. As you can see, for example, here, ELISA interaction forms inside of the two gold mirror. And then once laser is generated, a signal will be significantly amplified. And then we can generate a lasing signal and identify the changes of the ELISA interaction. And then here we also show other example of the peptide and protein detection. For example, we have demonstrated a lasing in human whole blood earlier. Nearly, we actually developed the first whispering gallery mode based laser for biodetection by developing a mechanism of using thread on the cavity interface. As you can see here, donor molecules are in the inside and the acceptor molecules on the outside. And then what we achieve is that we can identify molecular binding events once the molecules are attached to the surface through lasing signal. So as you can see here, as more molecules are attached to the surface, the lasing spectrum will also shift very significantly, and then we can see the wavelength shift very easily. In the bottom, we show another type of sensing mechanism. We use some droplet and then coat some protein and lipid on the surface. And by taking advantage of this FP cavity, we can identify and detect some like a molecular folding, a protein, a conformational changes inside of the cavity, and then convert it into some laser barcode for detection. And here we would also like to identify, especially emphasize that we can not only detect very large protein molecules, we can actually detect very, very small molecules, for example, like a peptide. I think somebody maybe turned on his microphone, so maybe you can mute yourself. Yeah, okay, so here we would like to show that we can also detect very small molecules by using the polarization property of lasing signal. Next, we move on to DNA molecule. So in the past, there are so many papers which has developed already by demonstrating how laser can be very sensitive to the binding event or the conformational changes of a DNA molecule inside of the cavity. So here we would like to especially point out the bottom right one is a DNA origami. So by controlling the geometric or the probes on the origami, the laser signal can actually be modified. So this is also one very important demonstration of using biolaser to detect DNA molecules. Moving from the molecular level to the cell level, here we show that how we can achieve lasing. So by creating so-called a cell laser, we can put a single cell or multi-cells inside of a two-mirror cavity. As you can see here, we can generate laser emissions directly by labeling the cells or the neurons. And then we can actually detect the changes, for example, the ion changes or the potential changes of the cell through laser emission. And then as we turn on or turn off some stimulus to the cell, we can also record the signals by lasing. The bottom one shows a very, very special type of cell laser, which actually does not even require two mirror. We take advantage of nanowire, which can generate a nano laser, and then we put inside of the cell or internalize directly by the cell to do sensing. And then you can see that as the NaCl solution or the osmotic pressure changes inside or outside the cell, the lasing spectrum will shift back and forth. And then by taking advantage of lasing spectrum, we can also use this to monitor the environmental changes inside of the cell. Moving from a cell to other types of organism, for example, by bacteria. This year, we also demonstrated the first 3D printed droplet-based laser on a chip. So we can actually use this to monitor and identify bacteria interaction with drugs. You can see in the left-hand side, as the drug interact with the bacteria or E. coli will be dead. And then more, like for example, DNA components or chlorophyll will be released and then interacts with the surface of the droplets, and eventually the laser emission will be enhanced. On the right-hand side is an example of a virus laser demonstrated in 2019. So other groups have demonstrated by modifying some structures of the virus, they can also achieve laser for some viral detection. Moving from molecular, cellular, and then now we are looking at the animal level in vivo. So as you can see here, at the Gathers and Subert's group in England and Germany, they actually have even transformed such a WGM microlaser inside of cells and inside of real living animal. So for example, you can see here, because the heart or cardiac cells will contract, and then it causes the environment changes, and therefore the laser signal will also change. So taking advantage of this, they can use it to monitor, like for example, the heartbeat of inside the fish, or even some drug interaction. Here is a video by them. So you can see that this bead inside of a cell, which is inside of a living animal, as the environment changes by different drug, for example, the lasing spectrum may shift slightly. So this is a way which we can conduct sensing in vivo level. All right, so far we have already demonstrated how this biolaser can be used for sensing. The second application which I am going to introduce today is how biolaser can be used for imaging. All right, so there are two strategies for achieving biolaser for imaging. The first strategy is to take advantage of scanning-based laser technology. For example, as we mentioned, we can achieve laser by putting the cells inside two mirror, and then laser can be generated, as you can see here. However, this is only limited to a single cell level. If we really want to form a big scanning image, we can use like a gavel mirror to conduct scanning laser, and then we can form a laser-based imaging. When I say laser-based imaging, I'm really talking about that the imaging, the signals are formed by laser emission, not fluorescent emission. All right, that's the first strategy. The second strategy is, for example, what if I do not have two mirrors? So scientists have actually taken advantage of the whispering gallery mode, which is, for example, they call it lasing particle. So such tiny lasing particle from micron to nanoscale have been developed, and then they can actually internalize such a lasing particle inside of the cells. And then they can track the cells. And then you can see here, for example, if I have two neurons, I have three particles with different emission color or emission wavelength. Then I can also conduct lasing imaging, for example. So this is the basic tool strategy of how laser, biolaser can be done for like a laser-based imaging. So the first one, as we already discussed, is called cellular-based biolaser. You can see here as the cell is confined inside the two mirror. You can see in the center picture, there are many, many beautiful laser modes generated from different cells. All right, so the center image shows a laser image from multiple cells. It's not single cell. All right, so these laser modes actually represent some structural information of the cell itself. This is very complicated and actually very interesting phenomena here. So there are many, many things which can be dig out in the future, possibly, because this part, there are still many unknown features until today. The bottom one shows that this technology can also be applied to, for example, different type of cells. So not only spherical one, it can also be applied to adhesive cells. For example, like HeLa cells, neurons, and as you can see on the bottom, there are many, many complicated laser images, like the flower, etc., at different wavelengths. So this is a very, very special thing which laser image may provide. However, there are many information which has not been well explored and understand yet. As I said, the second type of strategy is to use some micro to nanoscale spherical structure and internalize inside of cell. So in 2015, Andy and Humar, they have actually developed microbead laser and internalized inside of a single cell. And as you can see here, the bead, which is the green one here, actually serve as a laser itself. So each single cell can generate laser emission directly. We don't need any mirrors. This single cell can serve as a tiny laser. The bottom one is a more advanced development in 2018 to 2019. Instead of using some polystyrene bead or some oil droplet, they continue to advance the technology by using some quantum well and semiconductor material. So therefore, they can squeeze down the size from like a 5 micron to less than 1 micron. So now the cells can embed these tiny laser particles very easily, which does not occupy the cell volume too much. And then you can see here, they can track laser emission inside of a cell. So once the cell has 3 particles, as you can see here, 3 particles, it will correspond to 3 distinguishable, identified laser emission peaks. If the cell has 6 particles, then it will generate 6 laser peaks, for example. The bottom right shows another example of using plasmonic. So 2017 group has used plasmonic to develop some nano laser and also internalize inside the cell to conduct imaging. So this is a way of how this cellular-based biolaser can be conducted for imaging. All right, so taking advantage of this tiny laser particle, here we are going to introduce you some very important application, which is tracking and tagging. As you can see here, in 2015, different cells, because they internalize different particles. So therefore, by tracking the wavelength, you can actually distinguish which cells is which one according to the laser spectrum very easily. So this is a way of how scientists have used laser particle to track the trajectory and the movement of cells. They have further actually used this thing, and they actually inserted inside of blood and, for example, and inside of a mouse and cut out the tissue to demonstrate that this thing can actually go inside of a real animal circulation body and then identify the laser peak easily. In the bottom right, recently, this year, a group from China, they actually modified such lasing particle by inducing some scattering medium and scattering points on top of it. This is to resolve and to overcome one challenge in the past. The challenge is that sometimes the laser particle is only one direction, which is not easily to be identified. So this group, they actually identify a new method to track the lasing particle with multiple directions. Here, as you can see, more examples of using tracking in a tumor spheroid. So this famous work done by Harvard, they actually internalize this lasing particle inside of a tumor. As the tumor grows bigger and bigger through different hours, they can record the laser peak and the wavelength very easily. On the right-hand side shows that they can identify different motility, for example, low motility or high motility through this laser particle. So this is another way of how this laser imaging can be used in a tumor spheroid level. All right, so since we already talked about imaging from a cellular level to a tumor spheroid level, now we are more interested, is it possible to imaging at the tissue level? So in 2017, which is my PhD work, we developed so-called laser emission microscopy for human cancer tissues. So as you can see here, we put and cut out human tissue, especially lung cancer tissue, and label specific protein and dye target of interest. And then we sandwich the tissue inside the tumor, and then we can achieve laser emission by scanning across the whole mirror. Then we can form a laser image very easily. The reason why we do this, as you can see on the right-hand side, figure B, compared to conventional fluorescence, this technology will provide you a higher resolution in terms of spectrum and as well as in terms of spatial resolution. Yeah, for example, like here, we can see a blurry fluorescence compared to the bottom one, a very high contrast resolution laser emission. The reason how we conduct this laser emission microscopy, you can see here, both cancer tissue and normal tissue, they both can generate laser emission. However, the lasing threshold is different. So by using lasing threshold as a parameter, we can actually distinguish among cancer and among normal tissue. So this is how we identify cancer and normal by using this technology. So on the right-hand side, as you can see, figure F and figure I, if my pump is only about 24, laser will only be generated from cancer, while the healthy one will have no signal at all. So this is a way how we can do laser screening. So here we use this technology, and then we develop so-called high contrast early stage cancer diagnosis. This work is only limited to human tissue. Later on, we also expanded into animal tissue to approach some early stage cancer detection as well. All right. Here I would like to mention that besides using this laser for imaging in tissue directly, actually, this laser can actually also be combined with other important common imaging modality. For example, Raman imaging is very popular. So Gather's group has actually internalized such WGM laser inside of cells and tissue, and then they demonstrated that this can be an alternative way to sharpen out the signal for Raman and conduct a better Raman scattering imaging as well. Also, in 2020, Professor Fang's group from Michigan, they have actually also integrated this laser with OCT, which is optical coherent tomography. OCT is a very, very old and well-established imaging tool. However, they develop some tiny nano laser, which can be internalized inside of the cell and implanted such cells into rats or mice eyes for cell therapy. The reason why they do this is that the laser emission generated from these tiny nano laser can serve as like a light bulb to enhance the contrast agent and the surrounding environment. So this method can also improve the contrast and the imaging for OCT. So here I just introduce you guys the combining application with laser with some other very common imaging modality. However, most of the so far mentioned laser imaging, they are mostly done in the visible region. We all know that actually the biological window for human, for animal, for tissue, for cells is actually located around in the 700 to 1500. As you can see in the top image here, there are several near-infrared biological window, which is actually more useful for optical imaging because the light can penetrate much deeper into the tissue without have to sacrificing from the optical loss of human structure. So therefore, by using near-infrared probe as a pump is very important. So upconversion is a very, very promising material which can achieve near-infrared pump but visible detected imaging. So here we have show two examples by other groups. They have actually developed some whispering gallery mode laser by using upconversion nanoparticle. So they can pump by using, like, for example, a 980 or 800 nanometer laser, and then they achieve a laser in the visible region. Here they also demonstrate that they can put such micro laser inside of a tissue, especially human blood, and then they can monitor how the protein interacts with the laser. So as you can see, when a protein molecule flies across the WGM laser particle, the signal of the imaging will also changes. So this is another example of how people have been trying to advance the technology at different wavelength and different applications. All right. Okay, so since now we have already introduced about sensing, imaging, and now we are going to talk about bioinformatics. So one thing very special about such laser spectrum compared to fluorescent is fluorescent gives you a very strong signal in terms of very broad peak. However, you cannot identify any tiny feature because there's only one very, very broad fluorescent peak. However, laser or cavity modulated emissions, they have a very important feature is that they can provide very sharp and highly distinguishable peak. As you can see here, laser emission peak, for example, or modulated emission peaks, they have different modes. And then we can convert this mode into some barcode. And then this barcode, according to the intensity, or according to the peak wavelength, or according to the full width at half maxima, we can convert this barcode into like a digitalized like a barcode. But however, if you consider more complex intensity or other parameters, this barcode can be even higher capacity and even more complex. Okay, so the concept of using biolaser for digital information is we convert this very narrow line width part of the spectrum into barcode. And then we can use this barcode to identify different things or different changes. All right. So for example, by using this optical barcode, we can use it for molecular interaction. First, let's look at the bottom one. This one, we take advantage of a whispering gallery mode microsphere. As protein bind to the surface of this sphere, the interfacial energy will change, and then the spectrum will also change. Since the spectrum change, of course, the barcode will also change. So we take advantage of looking at this barcode, we can actually identify the molecular interaction on the surface of a microcavity. All right, if we're looking at cells, let's look at the top image. If a cell has one microsphere internalized inside of the cell, we can also convert the interaction or the information of a cell into a barcode. In the right-hand side, you can see this very fantastic image drawn by Institute of Physics. They actually show how this WGM sphere inside of a cell can be developed and converted into different barcode. So taking advantage of this barcode, it can be potentially used for monitoring cell interaction in the future. All right, so next we introduce something which is not very related to cells or molecules. However, it's also related to some healthcare technology. Here you can see that taking advantage of this barcode, scientists actually print these microsphere on top of some drugs. And then because under UV illumination, the barcode will change. And then it can be used as application in terms of anti-counterfeiting. And the second one, as you can see, a very famous picture. This is conducted, for example, by using some inkjet printing method. However, from far field, you can see this image. However, if you enlarge and zoom in, you can identify actually each pixel or each droplet is actually very different. And each droplet can be converted into a very highly distinguishable spectrum. And then this also gives you a way to do some anti-counterfeiting application. Yeah, all right. So based on this concept, scientists actually have also used this laser for some high security and some high security communication. For example, on the left-hand side, you can see scientists have actually fabricated some nano stripe and then put some dyes on top of it and generate very, very random distributed laser pattern. And then they can use this for some information encryption. The center one shares a very similar concept. However, they can convert this into some information unit, for example, 0, 1, 2, or 3 for some communication purpose. The right-hand side is a very recent work done by Professor Zhao in China. They actually use silk. Because silk, you can imagine that silk is very sensitive to its environment, pH value, whatever. So this biological changes can also induce some internal changes of the WGM structure. And then therefore the laser emission will also change. So by taking advantage of this complex interaction of the laser, they also use this as a physical unclonable function as a label to label some goods for anti-counterfeiting. So these are just some rough application which has been done in the past. All right, so moving back to some biological things. Here, I would like to especially mention a recent work done by our group. All right, so as I mentioned earlier, the laser mold or the laser pattern generated from cells are very, very complicated. It's very, very difficult to identify the real information which can be provided from single cells. Therefore, we come up with the idea by using artificial intelligence, deep learning method to help us understand what's actually happening inside of the cell. So here we use AI technology to study how different laser mode correspond to different biological information. And to our surprise, through this technology, we were very successfully able to identify different cell type. So for example, brain cells extracted from mouse, and then they were suspended. So actually their most important feature, which is the legs, are gone. They all turn out to be spherical, like cells. It's very difficult to identify which cell type is which cell. And through this AI-powered micro laser, we can predict and then understand what the original cell type was. For example, neuron or astrocytes, etc. So here is an example of using AI to help us understand, identify some cell information. Here we also like to introduce some biological control of the laser. So for example, recently we actually used DNA to control laser emission. So as I mentioned, that the DNA interaction inside of the cavity will interact with the light or liquid crystal. And then therefore the laser emission can also be controlled. For example, you can see in the center, through time at different seconds, the laser will shine and then quit and then shine and then close, shine and close. So this serves kind of like a logic gate application, which can be done in the future by using DNA to control some laser. Also, we actually use laser to investigate some brain network inside of a cavity. So recently we have a very interesting work by using the two-mirror cavity, and then we put some highly clinical relevant protein inside the two cavity, and then it forms very complex network. And then we discovered that this network structure actually can affect the laser output very dramatically. More importantly is that we discovered that as we monitor the laser emission, we discovered that actually it can correspond very well with some feature of the structure inside. So we also conducted some correlation metrics and found that actually this may be a potential tool to study some biological interaction in the future. Of course, as you can see in the center one, the 010101, of course, this may be possible to use as a tool to study some biological communication as well in the future. All right, so let's come to the very last and very important part, is how this biolaser can be transformed into a real world and real scenario application, especially on chip biomedical application. Okay, so here we demonstrate in our group by using printing. So printing is a very common technology nowadays, no matter it's 2D, 2.5, or 3D printing technology. We develop a new type of printing technology for laser biodetection. As I mentioned earlier, we can print E. coli, and then we can do some imaging to detect some interaction with the drug. In the bottom one, you can see on the chip of a mirror chip, we actually can form laser array. So laser array is a very important advancement to achieve high throughput, because in the future, we can do more like a larger dimensional scanning, and then we can do high throughput detection. On the right-hand side, you can see ELISA laser array. For example, as we mentioned, in 2014, ELISA laser has actually been demonstrated. However, it's only limited to like a single cavity level. If we really want to move this application to a real world, we need to have ability to conduct high throughput. So done by Professor Fang's group, by Dr. Tan, we actually fabricated, he fabricated mirrors with different micro-well structures in an array. For example, here we show a 2x2x2 array. Of course, in the future, you can create a larger dimension. And then we can create an ELISA laser by using this array. So this is just to identify a very important direction in the future, is if you really want to move this technology down the road, we need to integrate with some array technology to achieve high throughput application. Okay, so here, besides those two, we would like to introduce another two. One done by Professor Wu's group in China, Fudan University. Last year, he actually fabricated this toroidal-like microstructure, which serve as a micro laser. As you can see, as IgG binds to the surface of this toroidal-like micro laser, the spectrum will also shift. So they can actually achieve close to femtomolar detection limit by using this structure. On the right-hand side is actually done by Dr. Sun in 2015. Actually, in my opinion, this is one of the most earliest biolaser array on the market right now. So by taking advantage of protein, they actually created protein-based micro-laser array. So this is something very important, because instead of using silicon technology, actually, if you are able to use some biomaterial to fabricate array, this will be more biocompatible, and downstream in the future, this can be integrated with human and more biological tissue application. So this is, I would like to point out some array-based biolaser. Of course, besides array, people have also demonstrated a fiber-based array. So done by Professor Gong's group in China, they actually integrated several fiber aligned on a chip, and then they can scan through the fiber to collect the laser emission. Of course, this can give you a very high throughput, multiplex, or even multicolor laser emission for biochemical sensing. The right-hand side is a work which we call cell array laser or cell laser array. In 2017, we actually fabricated some micro well on the mirror and then cover the top mirror again and do some laser scanning. And then we can achieve this cell laser array by scanning through different points. And then we can detect the intensity. As you can see here, the yellow one shows the intensity of different cells, which corresponds to some different biological changes of the cell. All right, so now I am going to show a very recent work done by Professor Zhao's group this year. Besides using those laser array, whatever, on a chip, if you want to integrate such technology with human, usually on chip is very difficult. We need a flexible substrate to achieve good human interaction. Therefore, Professor Zhao's group, which is very specialized in all types of material, they actually fabricated such a micro laser array, but not on a silicon, not on a mirror. Instead, they fabricated on a polymer flexible substrate. In this case, they can actually wear this on the hand. And then when the hand bends, as you can see here, the laser will be deformed. And as I mentioned earlier, because laser is very sensitive to its environmental changes, structure changes, whatever. So therefore, as you can see, the deformation of the laser structure will cause huge changes of the laser intensity. So based on this concept, and they can actually control and monitor the different movement on the finger. So for example, this is a very, very good example of one of the first demonstration of human interaction with biolaser. In the future, if we want to integrate such biolaser with human, of course, fiber, especially flexible substrate and flexible fiber is definitely the best candidate, which I would say. So our group actually recently demonstrated multicolor lasing through some fiber. So if this concept can be integrated with some flexible substrate in the future, you can imagine that in the future, on the right-hand side, we may be able to conduct a multicolor, multiplex biosensing on a human for wearable device. All right, so there are some concepts very important to implement in the future. That is injectable, implantable, and wearable. In the first one here, fiber has the unique advantage of highly reproducible. So in the long-term future, we may be able to use a fiber, and then we may be able to use the fiber surface for biosensing, and it may be able to implant into human or animal to detect the changes. For example, like optogenetics. Implantable and wearable laser is also very important. As you can see here, Gather's group, they actually use some very lightweight polymer to fabricate this membrane-like laser. And then this laser can actually stick to your skin, or your nails, or even contact lens. So this is also a very good demonstration of some wearable and implantable laser. Here, we also show some other example. By using this whispering gallery mode, Humar actually demonstrated implantable laser by using tattoo. So actually the tattoo integrated with some whispering gallery mode laser can actually be implemented inside of a skin. The bottom one, as you can see from Professor Ta's group, they actually fabricated, developed a silica-based laser through random lasing-like mechanism. And then this, because it's silica, so actually it's more or less flexible, so it can be implemented on a skin. And the right-hand side, we show other example of some using flexible substrates integrated with some macro laser technology. Other groups have also demonstrated this can be integrated with human. All right, so the future of intelligent laser, I personally imagine that it may be injectable sensor inside the body for some health monitoring. It may serve as a light source for some phototherapy. It may be implantable or wearable and integrated with some AI, artificial intelligence in the future. So here I would like to introduce the concept of how we can reveal new information at the bio interface. As I mentioned, because laser is very complicated, it generates multiple parameters. So by taking advantage of the laser spectrum, laser threshold, laser mode, laser images, actually we can acquire many different parameters from laser, which allows us to conduct artificial intelligence and biointelligent laser information for cancer or, for example, neurological disease. In the future, I hope this field may be applied to some many other biomedical applications, from single cell analysis to precision medicine, on-chip devices, and even intelligent system. Lastly, I would like to share with you my personal viewpoints for some future challenges. As you can see here, biolaser, since the first development to today, this year marks the 10th year of biolaser. Biolaser, in order to become a real application or real device, it requires some critical points or critical challenges to be overcome. The first one is it has to be wearable, which means it should be implantable and flexible. The size should be smaller and smaller. And then, of course, this requires new materials to be integrated with biolaser. And more challenging, in order to be integrated with human, actually, so far, all the biolaser are optically pumped. In the future, if we really want to realize very good application, electrically pump is something very challenging, but very important. All right, let's look on the bottom one. So far today, most of the biolaser is just a laser signal. It may tell you some information, for example, like biosensing or bioimaging, but most of the laser, it does not provide deep information, especially functions. So in the future, we expect to develop multifunctional biolaser, especially not only in the visible, but most importantly in the near infrared region. Finally, in the last challenge, as I mentioned, laser signal is very complicated. However, nowadays, most people just use laser signal to identify, wow, laser. Okay, it's very strong, it's very sharp. However, it does not really provide any information for healthcare or biological technology. So therefore, we rely on some deep learning or artificial intelligence in the future to help us understand and to help us reveal the real useful information about health technology that can help us understand and identify human disease. So these are some future challenges which I would like to identify today. Finally, this slide I have shown 3 years ago. To my personal opinion, no matter currently if you are a PhD student, you're a master's student, or if you are a postdoc right now, laser has 3 components: pump, gain, and cavity. The pump usually is your supervisor, your personal aim, and your personal motivation. And also of course, this is something which is externally will trigger you, or internally trigger yourself to move forward. The cavity is something which will enhance your personal feature and interact together. So which means your current research topic and your lab resource, and especially your peers. Your peers are very important, especially in the peers in your lab or the peers working in a similar area. you may be beat by your peers or you may overcome your peers. The peers may form some very good interaction with you. So this is what I call the cavity. The gain actually is something inside of yourself, which is your mind and your attitude. Do you have a really good attitude toward in the future, no matter you will become a professor or you will become a PhD? Do you have good research plans beforehand? When you approach your professor or your supervisor, when you do anything, have you actually think about it? And do you have a real good research plan before? So I would say if you can maintain all these things, I anticipate you guys in the future can become very strong intensity like laser and become proficient like laser. We don't need to be like fluorescence. Fluorescence is very broad, which covers a huge spectrum, but instead we want to be laser because laser is very proficient. It's narrow line width and can give you very sharp signal to identify your personality and your profession. Finally, lastly but not the least, I would like to thank Professor Sherman Fan from University of Michigan. He is the guy who introduced me to the field of biological laser, and most importantly, my peers when I was a student 3 years ago. And then during the last 3 years, I am very fortunate to have many lab members from different areas on the world to work with me and to conduct all these research to nowadays. I would like to thank IAS, Institute of Advanced Study, to giving me this opportunity to share with you guys, and the funding support from Asst and Nanyang Technological University. And finally, thank you for all your attention today. I'm very happy to answer any questions you may have, or you can write email to me later on in the future. Okay, thank you.
Thank you very much, Prof. Chen, for the very interesting talk on biolasers, and especially the ending motivation that encouraging us to be strong and proficient like lasers. I think that's something that we graduate students can associate really well with. We now move on to the question and answer segment of the webinar. I encourage students to post their questions in the Q&A section at the bottom of the screen. You may also ask the questions by raising your hand. You get to interact with Prof. Chen directly. So please use the raise the hand feature at the bottom of your screen and we will unmute you so that you can ask your question directly. Before we go on to the questions in the Q&A section, we have some questions in advance here. Biological lasers require injection of some kind of material or particles in, for example, animals or humans. Are there any long-term impacts of having these kind of particles in the body? Is it safe to have these kind of particles for a long time inside any biological being, be it a human or an animal?
All right. Yeah, so this is a very good question. This is why we require biological material-based laser. Because, for example, if you use some toxic material, it may not sustain a long time in human body. So there are many groups which tries to use biomaterial laser, which can stand long term and have good biocompatibility.
Thanks, Prof. Another question is related to the laser's performance. So when these laser particles or lasers are inside a biological being or organism, then how about its performance? Does it degrade over time? How do we ensure that an implanted laser continues to meet expectations and give the kind of information that we are looking for?
So you're asking about the degrading of the signal? You mean the intensity?
Yes, the performance, yeah, the intensity of the.
All right, lasers. So I think as I just said, many groups, they are trying to develop laser particles. So especially many groups, they have tried to demonstrate they can stand a long time. They try to pump the laser for very multiple, like 1,000 million of laser pulses. And then they demonstrate that the signal can be very consistent. Yeah, so I would say organic dye material, this may be the weakness because it may bleach. However, maybe some other, for example, perovskite, semiconductor material in the future, it can sustain better, preventing this bleaching behavior. Yeah.
Thanks, Prof. I'll now take on a question from Hong Chuan Lim. He has a question asking, what are some career opportunities in this field of biolasers?
Pardon, can you repeat again?
What are some career opportunities in this area of biolasers and related areas?
You mean career?
Yes.
In career, you mean like maybe, okay. So career, I would say, if you're talking about academic career, I would definitely say academic. We welcome everybody to join us because this is a field which is more or less quite new. If you want to be a scientist or professor, this may be a good career. In terms of in industry, I would say you may want to check out a startup company spin-off from Harvard University called LASE. Based on the laser particle, which I introduced today, they actually, I think, have actually attracted a big market for laser particles. So by using this laser particle, you can imagine that fluorescent dyes and quantum dots, right? They have already go into the market and earn a big fortune. So I believe this laser particle related thing may give you a good opportunity for industry in the future. Yeah.
Thanks, Prof. There's another related question to what was asked earlier. Is there any research done related to biotoxicity or durability of the current biolasers?
Yes, definitely. In most of the papers, as long as they have cell experiments, usually the reviewers will require us to conduct at least 7 days toxicity experiment to demonstrate the vitality of the cells. Yeah, so this part has been considered carefully.
We have one more question from Hongbo. He wants to know, how can we understand the intelligence of biolasers?
Okay, so this topic is very interesting. I call it biointelligence. The reason is you can imagine that as we do biosensing, you detect the signal and you acquire the information of the biological substance. On the other hand, you can actually imagine it as an active way. If the laser signal can directly tell you some information about your disease, about your current body healthcare. Yeah, so this is why we call this biointelligent, because this laser can actually interpret some information about your body potentially. Yeah.
We have a question from Gayatri. She's curious to know more about biolaser-based imaging. So fluorescent signal-based imaging techniques are quite advanced today and give high-quality images. She understands that the laser gives sharp spectrum signature. However, that is spectral analysis. What is a particular advantage in imaging using biolasers?
You mean the advantages of using biolasers?
Yes.
All right. I think the first one is that the biolaser gives you a very, very strong intensity. So this part can solve many issues regarding like a low contrast or low signal to noise ratio, especially when the background noise from, for example, like fluorescence sometimes suffer from background noise. This can solve this problem. The second one is especially, for example, like the spectrum. Because the spectrum is very narrow, so if you conduct spectral imaging, I would say this may be a very good application by using different spectrum to identify specific features directly from the image. The last one is multiplex, because you can imagine that the line width is very narrow. So within the same parameter, for example, 100 nanometer, we can put, for example, like 20 to 30 colors. And then you can conduct multiplex imaging very easily compared to fluorescence.
We have a question from Adrianska. He wants to know if there are any theoretical applications of electrical powered biolasers. How will these lasers be powered? Will electricity source be internalized to the biolasers, or will there be a need for wireless charging from wearable devices?
All right. As I mentioned, that electrical powered is something which most people working on laser are pursuing, because that's something very important to move in a real world application. However, based on my knowledge, there are no electrically pumped biolaser so far.
Okay, thank you. We have a question from Wang Jamin. His question is whether the power density of the pump limits bioimaging applications, especially in living systems.
Pardon, can you repeat again?
If he wants to know if power density of the pump, does it cause any kind of limitation for biological application, bioimaging applications when we use biolasers?
All right. Okay, so the first one, maybe I'm not sure if he or she is considering about, will the laser power damage the tissue? So far, because the laser power which we use are at least 5 to 10 times lower than the surgery. So therefore, this may not be an issue. Additionally, we use pulse laser, so this may not cause damage. In terms of the power, I would say it depends. It really depends on what material, what gain, what type of structure, what type of cavity you are using. If you have very, very good optical cavity structure and very, very good gain material, and therefore you can achieve laser with very, very low power or low energy. However, if the cavity is bad, the gain is very worse, then you will have to require high power, high energy to achieve laser emission.
Thanks, Prof. We have one more question. It's related to what is bleaching mechanism and the speed of biolasers? Can it be used for long-term monitoring?
Long-term monitoring, yes. One of the slides has shown that in tumor spheroid, so far as I understand, they can monitor up to like 72 hours, 90 hours with no problem because the laser is very stable. The laser is semiconductor based. That one is very stable, which can survive long-term imaging. However, if you consider those organic dye-based laser, no. So far, they bleach quite fast. It does not survive very, very well for long-term imaging. But that's something which we will definitely try to solve in the future.
Thanks, Prof. If any of y'all have further questions, y'all can continue to post the questions in Q&A. We will keep this channel open. In the meantime, Prof, I have some questions related to the future of biolaser, especially when in the direction of wearables and for human applications. So I understand that we are looking to something like fiber-based wearable biolasers. And these biolasers are quite sensitive to change in environment, so we should be able to detect the intensity, variation in the intensity, and accordingly detect some abnormalities underlying in the skin or below inside the human body. So will there be a different signal even if there's an interference between multiple factors which are affecting the laser to react or to spike at a certain point? Like, how do we distinguish if it's coming because of a particular cell inside a body or from another source which is interfering to cause this laser to fire up?
All right. I think this is a very good question, which is very in-depth. Yes, it is important to identify which is your signal related to your biological substance of target of interest, or it's just due to some scattering or some other medium. An easy way is to label the target. You can look by color, for example, or the emission wavelength. That is a possible way. Another way is the calibration is very important at the very beginning, especially having some specific binding pre-process. Yeah, I think this part can avoid some issues which you have mentioned. However, as you mentioned about the human part, that part is too advanced. I'm afraid I cannot tell too much about that, but in human is much more complicated in the chip-based structure. So of course, these are down the road which we have to pay attention and we need to overcome.
Thanks, Prof. Thank you very much. I will pause for another 2 minutes or so to see if you have any further questions coming up from our audience. If not, we can wrap up for today.
All right.
We have a question from Gayatri. She wants to know, how do we envision to pump these lasers for wearables in the future?
Okay, so if you want to use optically pump, of course, for example, if it's a wearable device, you need like external something to shine on your hand, for example, like scanning. So it requires another device, for example, to perform the pump. So for example, you wear a watch and you need to move closer to a certain region to excite your watch. However, as I mentioned, if it's possible to develop electrically pump laser in the future, then we may not require external device for the pumping. Maybe we can, for example, just turn on, turn off, and then you can detect with your wearable device. Yeah, so this is very challenging and something very interesting.
Yeah, that sure sounds interesting. Thanks, Prof. We have a question from Nabila. Apart from imaging, can lasers be used to reduce tumor size?
Tumor size.
To reduce, can lasers be used for treatment, basically, is a question.
Oh yeah, sure. One of the Nature Communication paper I mentioned with the plasmonic particle, they actually use phototherapy and thermal therapy. Yeah, because one thing very important is laser has very strong intensity. So actually it creates heat very easily, or creates very strong light easily. So that paper actually, you take advantage of this heat and then to enhance some image contrast, and then also take advantage of the light to cure some cells inside, especially cancer cells. Yeah, so it is definitely possible to use the tiny laser and implement it into tissue for some photodynamic therapy in the future.
So in the future, we could use lasers for both detection as well as for treatment of.
Yeah, that may be possible, or even outside, not from the inside. Yeah.
That's interesting. We have a question from Yinxiang Liao. How do you tag the biolasers to specific modules? Is it the same way as you tag fluorescence modules?
Yes. Actually, most of the laser technology is based on the fluorescent-based detection method. Yeah. So whatever you can do to fluorescent-based, it may potentially can be transferred into a laser-based technology.
Okay. Prof, I have a question related to AI-powered lasers. So how do you create the data set or the ground truth? How do you know which cell be detected by the laser is what cell? Is it a neuron cell or?
Oh, actually, the cells are provided by SCBE, Professor Zhou Xinyan's group. Actually, her postdoc already prepared separately. So we actually already have the ground truth. We know which cell is which cell at the very beginning.
Okay. And I also have another question related to the experimental setup from the video that you had shown with the wearable glove and with the laser being emitted when the finger bends. So is there a camera recording the change in the intensity, or how is it detected, and then how is it processed after that?
Okay, so I think the most common way is to detect with a spectrometer.
Mm-hmm.
Yeah, because spectrometer will give you the laser sharp signal directly. However, because that work is not done by my group, so maybe I didn't need to look into details. However, I can say that by using CCD imaging camera is the future, because it's more easily to be detected, because spectrometer requires good alignment, while CCD imaging is more easily to be captured directly. So if it's possible to be collected directly through a camera, that will be more possible for beneficial for more application.
Thanks, Prof. I think that clarifies. From Tam Nguyen, is there any chance for electrically pumped biolasers? I think this has been addressed. Yeah.
Okay. Electrically pumped biolaser, one of my dream. I would say it's very difficult. First, you need to solve electrically pumped organic dye laser in general. However, there are papers which have demonstrated similar concept out there already. However, related to bio, there's not anything out there yet. However, if you consider electrically pumped quantum dot or perovskite laser as a source, that may be possible. And then if it's possible to integrate with some human protein cells, I would say that may be a good way to conduct a biologically integrated electrically pumped laser.
Thanks, Prof. I think if there are any further questions, you may contact Prof. Chen, or you may direct the questions to Ms. Chris or IAS, who can forward those questions to Prof. Chen. Prof. Chen, thank you very much for the very interactive and very interesting talk today. It was really
Thank you very much for hosting this, and also thank you for all the participants staying. Yeah.
Thank you.
Thank you.
Before we end the seminar, we have the attendance session now. Thank you all for your wonderful participation in this event. Before you leave, do scan the QR code, which will now be shown on your screen shortly. We value your feedback. The feedback form also has the attendance part in the end, and we will forward it to your respective GSEs and schools for the attendance endorsement. You can now see the QR code on your screen. Please take a few minutes to fill in the feedback form and give in your attendance. Thank you.
Okay, thank you.
Thanks, Prof. Chen.
Okay, bye-bye.
Bye.